Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show. Bring you another awesome guest today, involved in creating a better tomorrow for so many people out there. Today we have the honor of being joined by Joel Greshock, who is Vice President, Data Sciences Oncology at Janssen Research and Development, part of the Johnson & Johnson group of companies, where he's responsible for creating unique and actionable medical insights using their large, increasingly available data sets, ultimately with a focus on research that allows the discovery of novel therapeutic targets, identifying areas of unmet medical need, and then ultimately enhancing their clinical trial recruitment and execution. Prior to joining Janssen, Joel served as Vice President of Bioinformatics at the startup Neon Therapeutics, where he was involved in building and managing their data sciences organization, ultimately responsible for the design and the deployment of their personalized cancer therapies. Prior to Neon, Joel served as Head of Oncology, Translational Informatics at Novartis, responsible for the correlation of patient outcomes from molecular biomarkers, identifying mechanisms of clinical resistance, computational research for their various assets that were either in clinical development or are being evaluated for clinical development. And then prior to Novartis, Joel was involved in numerous roles for GlaxoSmithKline Oncology, including Head of Bioinformatics. Early in his career, he was a data analyst at the Abramson Family Cancer Research Institute here at the University of Pennsylvania down the street, bringing and building early generation microarray platforms, developing predictive models for cancer predisposition mutations. Joel holds a Bachelor of Science in Comprehensive Science and a Master's of Science in Biology and Biostatistics, both down the road at Villanova. He also completed studies at UPenn and Temple in statistics and computer science. We're honored to have him with us today. A lot of really interesting themes to get into today. Joel Greshock, thank you so much for taking the time to come on the show.
Yeah, thanks, Ira. It's great to be here.
Yeah, Joel, I just love to start off because, you have this, wonderful set of intellectual interests, in biology, bioinformatics, computer sciences, of course. And, we were always told, 20 years ago about the important convergence of these tools that were coming together. Talk a little bit about the beginning, if you would. What got you interested in sort of this really unique area that's obviously taken off and blossomed substantially over the last couple of decades?
Academically, I was always interested in multidisciplinary science, but I always gravitated towards the life sciences as like an area to apply it. But life sciences were more traditionally like a data poor area of science. So there wasn't a lot of effort to bring in advanced analysis to life sciences, and so. But I knew that big data sets really are how you can really gain novel insights into disease and biology. And with the genome age, it just really kind of blew up the total volume of data in the life sciences. And I sort of was lucky enough to my career to come of age during the transition from the pre-genome to the post-genome era. And that was a really exciting thing to see. So I from a career perspective, it was just a very, I took sort of an opportunistic approach where, I started getting involved with some of the work that resulted from the publication of the genome. And it was just one of those things where like the genome was released and everyone was excited about it, but there wasn't a lot of knowledge about how to use it, both on a technical and scientific level. So I really, got interested in how we could apply, novel analytic techniques to the genome. And another critical advance that came with the publication of the genome was advances in like microtechnologies that allowed us to measure disease or like nucleic acids or proteins at the genome level. And so when you coupled the genome and being able to map the genome with genomic assays, it led us to be able to characterize disease in a really unprecedented way. And I'd say that there was a very rapid advances in the early 2000s that brought us into this genome and genomics age, and it's a really exciting time to be in the field. So I think that's where my personal interest in bringing like advanced statistical techniques to the life sciences really got more fleshed out. So anyway, it was an exciting time, and that's sort of how I sort of got my early start in bringing data science into life sciences.
And continuing along that, as we chatted a little bit before the show, I spent your time sort of swimming through, your extensive publication history, in the peer-reviewed literature, and looking at, while you were here at Penn, you were studying breast cancer tumors using oligomicroarrays. You had published on SNP polymorphisms and immunomodulation, and this was sort of at a time where, you were mentioning some of the private initiatives were beginning to take off. The Human Genome Project had a couple more years to go at that point. Talk a little bit about, because obviously genomics was the hot term then, a lot of other omics and thinking about sort of the data that's beginning with the transcriptomics and proteomics and all the other omics dumping exponential amounts of data in your lab, plus the support tools, the AI and all the computational bioinformatics stuff. Talk a little bit about how you've seen the evolution and convergence of all these tools over those couple decades, per your experience at the as I mentioned in the bio at these various companies.
In the early days, everything was very do-it-yourself. There wasn't a lot of off-the-shelf, assays, commercial assays or analytic tools available. So people who were in the field, had to sort of roll up their sleeves and construct assays. So, one thing that I did that you mentioned was really got involved with the construction of early generation microarrays. The area that I was focused was on cytogenetics, and getting a higher resolution picture of genomic copy number changes, and the focus was cancer, but it was really something that could be applied to any disease area where you could really map small cytogenetic changes and unlock disease drivers, and so but it wasn't something that you could just go and buy an assay and analyze disease tissue at scale. We made them ourselves, and then the accompanying analytic tools were also something that very much had to be homegrown, and I think it was an area where we sort of started connecting knowledge gaps with what was happening in the cancer genome, with these tools that can really give insights to fill the gaps. So in the lab side, it was really doing things at scale, culturing things like bacterial clone libraries, mapping those to the genome, constructing assays. Then on the bioinformatics side, developing things like limb systems and tools to visualize data, derive insights from panels of patients. I'd say that the evolution of that over the years, and obviously there's been a lot of advances in assays since then. So, looking at, genome-wide copy number changes at a 1 megabase resolution seems like old news, but at the time it was new. So I think there's been a lot of advances in assays such as NGS that have really taken this to a new level. And I think there's been a lot of commercial efforts to bring in really nice analytic tools, and then public and private, tools that have really, enabled better, resolution and understanding of some of the, disease-specific changes that we see in the genome. So, in hindsight, we really sort of tore down a lot of barriers and sort of set the table for, what has kind of advanced in industry, in academia since then.
Excellent. Let's introduce at this point your team and sort of the data sciences oncology group, because as I mentioned in the bio, a lot, a rather broad purview. So discovering novel therapeutic targets, looking at other sort of unmet medical needs, potentially per existing drugs, and then obviously the whole sort of clinical recruitment. Talk a little about sort of the team, its construction, what typically goes on, and sort of where the directives come from, because obviously I can see, some of the folks in the organization working, developing sort of early stage candidates need your help in maturing the targets. You might get contacted from some clinical research associates. Hey, I need your help in recruitment here. Talk a little bit of how that works, and then, sorry, there's a long question, but third part of that. Clearly, you're heavily focused on oncology, but Janssen has immunology, it has cardiovascular, a lot of other areas where maybe proliferation may come into play, whether it's things like fibrosis or hyperactive immune response. Any interesting sort of cross-fertilization of the tools you're developing for oncology in some of the other J&J adjacent therapeutic areas?
Yeah, of course. So the data science organization at Janssen is a very multidisciplinary team, and so my specific team focuses on oncology, and really connecting the dots between where data science can be impactful, on our portfolio, and really being able to bring in the technical expertise to then really deliver solutions. And so the organization is sort of split into two kind of like general areas. One of them focuses on the therapy areas. Another broad area, component of the team is really bringing the technical know-how, whether it's AI, machine learning, data platforms, clinical background and clinical development, as well as real-world evidence. And so, we really have four key pillars that we focus on broadly. For us in oncology, that the priorities fit into these key areas. So, one of those is really focusing on patient identification and stratification. So how can we really use data science approaches to better, identify patients who may benefit from therapies? And so for that, we work closely with our companion diagnostic colleagues, translational research in oncology. Another area focuses on real-world evidence, and building evidence to support clinical trials, whether it's in the trial design phase or really at the time building evidence to accompany regulatory submissions. Another related area is really using real-world evidence to operationalize our trials. How can we really identify the right kind of clinical trial sites? How do we match patients through these just-in-time networks to our clinical trials and really run fast, efficient clinical trials using data science approaches? And then the fourth pillar really comes on the early discovery side. And, how can we better identify targets using data science approaches? And there's many different ways to do it. But really on the early side, how can, picking targets is one of the most important, exciting things that we do. And using data science to really triage and evaluate targets can identify higher quality targets and also replace even some of the early, typically through low throughput assay evaluation of targets to really help fill our portfolio. The team itself, is very multidisciplinary. We have people from, engineering backgrounds, more traditional computational bioinformatics backgrounds like myself. We have physicians on the team that are really bringing together our clinical science and data science. And we need those. We need that multidisciplinary team to make it all work. And so it's that way by design. And so that's really like how we've been able to really get integrated with all the therapy areas and be part of the team. I mean, as an organization, we're still relatively young, but we've done a lot of work to really make sure that that the data science team is just so very fluidly integrated with the clinical development teams at Janssen, the discovery teams, and we've got a seat at the table. So to address your third question, there's plenty of opportunity for cross-fertilization. And some of this is mechanical, like just say, how do we license data sets that really provide insights across disease areas, or thinking about it scientifically, immunology, we think of it, although it's thought of as a sort of a discovery, like a therapeutic area here, as a, it's a system that really touches all disease, and that's cancer, baby being one of the biggest, areas where immunology has had such a huge impact on how we treat cancer in the last, decade or so. So that's always something that's front of mind for us.
Excellent. Let's talk about a couple of the examples, Joel, because as again, you've been active publishing, and I thought an interesting place to start per some of these themes would be your I believe it's a 2020 paper in Trends in Cancer. It was entitled "Harnessing Real-World Evidence for the Development of Novel Cancer Therapies." And it's quite interesting because here in this paper you outline a lot of the challenges associated with novel therapeutic development and the real world. And here in 2023, a couple of years later, but some of these technologies, like one of the things you talked about were sort of the high value data fields that were unable to be captured at that point. And you talk about natural language processing, which, pretty hot theme the last couple of months. You talk about machine learning in terms of the ability to, again, match patients in this paper. Take us into the topic of real world evidence per an organization like Janssen Oncology, and a little bit of some of the ways you can utilize these new tools.
Sure. I mean, real world evidence is an area that could be, it has a growing level of influence around how we develop drugs. And I mean, it could do everything from speed the development process to better identify patients who could benefit from therapy. So we're really excited about real world evidence being a key part of how we think of drug discovery and development in the future. But it's also an area that's riddled with challenges too.
Sure.
From our perspective, I think that everything from like the discovery process, we're integrating real world evidence into evaluating areas of unmet medical needs. So could we take these multimodal data sets where you can combine cancer genetics with patient treatment, paradigms and outcomes to say, look, it's really these segments that are not benefiting from current standard of care. And even think about how we discover and design new therapies around those patients that have high unmet medical need. And then subsequently designing trials around them. So like, for instance, could you take real world data, look at things like, inclusion, exclusion criteria from a clinical trial, understand that, how these patients are being treated. For example, a disease like multiple myeloma, there's a lot of treatments out there that have emerged in recent years. And, understanding what standard of care is, even regionally, is something that we turn to real world data to understand. And I mentioned earlier things like site selection. So, bringing in data from across, say, the United States and understanding, where patients that may really benefit from these clinical trials are being treated. One other area that's just been incredibly important for us broadly is really getting, improving the level of diversity and representation of patients on our clinical trials. And part of that is really understanding where and how these patients are being treated, so we can really bring the trials to those patients. So we have more representation of things like ethnic and racial minorities on our trials that really are critically important in evaluating how these drugs are working. And you mentioned earlier, like recruitment, there's more companies, an example of this is Tempus, who've got, networks of clinical trial sites. And what we do is work with them to use real world data to evaluate, patient eligibility across their network and see if we can link patients using, their medical records to clinical trials. And those are areas we're all really excited about. Maybe Ira, one last point I'll mention we're always interested in advancing how patient outcomes are evaluated. So real world data is going to become important for how we look at endpoints. So can we develop better surrogate endpoints for things like overall survival that you can get a faster readout on clinical trials to advance decision making in the drug development process?
Absolutely, absolutely. So another area that you mentioned when we first sort of started to discuss the show that you were quite passionate about was the topic of imaging, and in this era of personalized medicine, sort of the detailed imaging of specific tumors in terms of recognizing them and measuring them in terms of response to different therapies. In addition to your responsibilities for the data sciences team, you're also involved in something called Project Data Sphere, the Images and Algorithms Task Force, where you're, I guess the J&J representative amongst this group of radiologists, oncologists, machine learning specialists to really improve exponentially sort of this imaging component of personalized oncology. Talk a little bit about this role, if you would.
Yeah, I mean, so I am very excited about what we can do with medical imaging. And just to take a step back, we've been collecting medical imaging data for, in oncology for a long time. So whether it's like, pathology to help diagnose patients and understand the etiology of their disease, to radiology data, we're required to capture that for clinical trials. So, one of the reasons for the excitement is that we've been collecting imaging data for many years, and we have a lot of it. And it's only more recently, has the field of machine learning and AI been able to really, tap into the imaging data, to gain novel insights. So there's been a lot of methodologic advances recently that have been able to, uncover things that can't be done through manual evaluation. And so, things like, and there's like certainly a lot we could do with it. One of them is really, automating how we evaluate disease. So for instance, on our clinical trials, we use a protocol called RECIST 1.1 to report how a patient disease is, changes after treatment. So like, to what degree does the tumor shrink or grow? And we report those are results that are commonly used to drive decision making in the drug development process. There's a lot of inconsistencies in how this is done from radiologist to radiologist. So it's an area that clearly, with some very, savvy machine learning AI could automate the task. And so, whether it's using it maybe initially for just to adjudication of differences between radiologist evaluation of outcomes, or maybe in the future just more the frontline evaluation of how patients respond to therapy. Certainly a great application. Another area with imaging that's just really exciting is with diagnostics, and for instance with pathology data, as I mentioned. Like, we routinely collect pathology data on all of our patients in cancer, in oncology. And I think there's a lot of opportunity to apply algorithms to things like H&E stains to identify some common disease biology that could be used as a diagnostic. So that's something that we've been working closely with our diagnostic team to evaluate opportunities there as well.
Excellent. Joel, one area that the FDA has been kind of active the last couple of years in putting out guidance on is the area of combination products, or combinations, polypharmacy, whatever you want to call it. And obviously, aside from sort of HIV, cancer has been one of those areas that sort of the concept of the combination and the ability to attack different pathways and so forth in therapy has been so crucial in taking some of these death sentence cancers to a different place. Talk a little bit, because I know you published in the past on sort of tools to look at drug combinations and understand them better, from the sort of the old method of, let's develop drug 1 and drug 2 and drug 3 and combine them at some point in the future. Talk a little about this from the perspective of the way you think about developing combinations for cancer, and potentially, the theme of repurposing has been kind of hot lately as well. Any interesting learnings that come out of your work in that area in terms of how you think about not just the novel therapeutics that you're developing, but interesting combinations per repurposing?
Sure. Yeah. I mean, combination therapies, I think, are going to be an important, approach to really developing curative regimens for cancer. I think in the earlier days, that the TCGA, CCLE type approaches where, we could do like these drug screens on tumor models, was fruitful, but it also didn't solve all the problems. So there was these brute force approaches to identify, what combinations are synergistic. I think one of the challenges is that the mechanism, for combination activity, there's not necessarily one common thread for it. So it's not always just, targeting two cancer pathways simultaneously, but it's rather, if you enter the immune system into it, it's like, how do you perhaps like, first lead with something that could reduce the size of a tumor rapidly, followed by a combination, like an immune therapy. So I think that the bottom line is that there wasn't one sort of silver bullet approach that found all the best combinations. So we're left with still a piecemeal approach, and some of it is also learning about how patients react to therapy at a molecular level. And I think maybe that's an area that really could be fruitful in how we think about combinations, both drug sequencing, in which patients should get combination regimens up front. One thing that we do is look at things like, minimum residual disease on patients on some of our trials. So you treat, and can you characterize how a patient responds to therapy by looking at, indicators that are at the molecular level, say they're circulating, in blood. And so, so now like we use those to say, look, have you eradicated disease at a molecular level, looking at minimum residual disease? I think in the future, as we advance assays and where you can look at not just how a patient's responding to see whether they have some disease burden or not, but it's rather, is there something specific if their patient has disease left over after treatment, what's unique about that? Or if a patient then progresses, after therapy, what's unique about, the disease burden that's progressing, and whether it's like, for instance, an example is with, EGFR therapies in lung cancer, one, recurring mechanism of resistance is activation of the c-Met pathway. And so, are there ways to determine which patients perhaps get combinations of EGFR and c-Met up front? Or are there ways to say, look, if you measure a patient's disease burden, in a serial manner after treatment, and you see early indications they may be recurring, what is the mechanism? Is the mechanism c-Met? Do you get, use that as an example, like post-treatment? And I think that, those assays will improve over time. And so being both, understanding which ones, should be given up front, what combinations, there's always toxicities managed with combinations, or in most cases, versus cases where, you could be reactive to how a patient responds. And then if they have progressive disease, how to intervene with that early, so you can drive towards a cure in that patient.
Perfect. Joel, we've talked a little bit about some of the tools that you've been using in the past, and then in recent years in terms of the natural language processing, the AI, and so forth. But we profile sort of a broad range of the future-facing stuff on the show. We've talked about in silico drug discovery. We've talked a little about the theme of digital twins. Talk about sort of what gets you excited in terms of these cutting edge, let me say bleeding edge tools that are on the horizon, and then a little bit about how you, per your responsibility of data sciences, evaluate, because I'm sure just like, you have small biotech companies coming into Janssen to pitch you on their compounds. I'm sure you got a lot of people trying to pitch you on these new technologies. Talk a little about the evaluation process.
Yeah, I mean, a technology that I think is going to become a staple in really how we develop drugs are single cell technologies. I think from the discovery side, through what we do development, even things like I mentioned with characterizing how a patient responds to therapy. And though these, I don't know if people, they're not brand new, I think in the scheme of their life cycle are still very new. And so, whether it's, disease profiling or building atlases of what normal healthy tissue looks like, this will really help us sort of contextualize, unique features of disease cells. So for instance, could we characterize, disease cells and to better understand how to apply and optimize CAR T therapies? Can we use single cell profiling as a tool to optimize our CAR T therapies for the future? And like I said, even understanding how patients respond, why patients may or may not respond to specific therapies, I think single cell will be a big part of that. And so I think it also, maybe another one just to reiterate, how we characterize disease burden in patients is this a really critical way about, with how we could drive towards curative regimens in cancer. So, if you have, if we've got novel therapies that can really lower or eradicate disease at the molecular level, how do you ensure that if the patient progresses, that you can catch it early when it's most discoverable and treatable, time using these assays? And I think that as those advance, we will get better at recognizing when a patient, might have a disease recurrence and be able to intervene in the right way very early on. So those are areas that I'm super excited about, and maybe just one mention on the analytics side. I think I'm extremely excited with what we're doing with like machine learning, AI approaches to data integration. So how could you bring multimodal data sets together, whether it's real world evidence, medical record data with genomic data, medical imaging data, and derive new insights from a diverse set of data rather than thinking of them in silos. So that's, I think, certainly something that we're making progress, but there's a lot of work to do in really true data integration. Certainly, there's a lot of emerging companies in the field of data science. Janssen has a rich history of external innovation. It's definitely a very important part for data science. We can't have a expertise on every technology internally, so we very much turn to the external universe for to really complement what we do internally. So we're always talking to new and innovative companies. And some of it's like to some degree matchmaking, finding an area that's a need of ours, an area where we may have some gap in knowledge, and building a partnership, the right kind of partnerships to really solve problems, and work together with these innovative companies. In some cases, these are larger, more significant partnerships. For instance, ones we have with companies like Tempus and Concert AI and Flatiron. In other cases, it's more focused on a single disease. For instance, what we do with this company Ultromics to improve the diagnosis of AL amyloidosis. Certainly, we want to continue with this model, and it'll be an important part of how we continue with the data science, impact with drug development.
Outstanding. Joel, so we've talked about the sort of cutting edge personalized therapeutics that you're developing at Janssen. You have the data sets, you have this amazing suite of technologies. Take us to the future now. Take us on a little journey of where you hope to be in 3 and 5 and 10 years from now, per how this is all evolving.
Yeah, I'm very optimistic, about the future. I think, we're just at the beginning here with the data science journey, in pharmaceuticals and medical research. So I think there's a lot of exciting things that I could highlight. I think, one area that I think will become much more impactful is how we conduct clinical trials. And so doing things like pragmatic trials that leverage real world data through networks of institutions to collect data across, for instance, surveys of how patients are being treated with standard of care, even addressing things like post-marketing commitments through pragmatic trials. Certainly a very exciting way to collect a lot of data quickly, to learn about, to get novel insights, and also, provide very valuable data to regulators. So I think that's maybe a thematic area that I would say is something I'm really excited about. I think, just broadly speaking in the field, right now this is a very much a collection of different fields that have come together. I think over time this will become much more unified. So, bringing together now, like, real world evidence and medical imaging, sometimes get thought of as being in silos, but as we advance and develop new talent in this area, and it becomes more mature, those two things will go hand in hand. And maybe some of it's just really like, whether it's like platforms that can integrate these data types, methodologies that can combine them. I think that will just become, I think that we'll think of areas of data science much less in silos and rather more unified. And maybe one other point I'll mention is on talent. I think talent will become much more fluid. Back in the old days, I mentioned if you had someone who knew some biology, genetics, and could do some Perl programming, they were like, kind of a unique thing. I think that, we've gotten by that. I think there's a lot of bilingual talent out there, academic programs that are really making concerted efforts to sort of break down boundaries, in academia. And I think that will continue. We see more physicians in the field who are really developing, talents in technical talents, an understanding of data science, and bringing their knowledge of medicine, which is critical for drug development, into the drug development process. And I think the next generation of leaders in this field are going to be, career multidisciplinary scientists, which is just really exciting.
Continue on that for a bit, because obviously 20 some odd years ago someone was like, some mentor of yours was like, Joel, you like biology, you like computer science, get involved in this bioinformatics stuff, it's going to be hot. We do have a cohort of our audience that is in that next generation. I make my 3 kids watch the show, although they roll their eyes at me, but talk to the next generation a bit. What advice would you give for that that college student, that high school student, maybe that's kind of interested in health, biology, whatever it may be? Where are you going to direct them in terms of the way all this is going?
I think the overarching message is, don't be afraid to break down boundaries, in disciplines, and really, think about, how to make an impact, and start connecting, like, what really, things need to be done to make that impact. If it means bringing, medicine together with data science, or technical capabilities, breaking boundaries by really recognizing that these fields need to be integrated is just, like, I think, a critical thing for people to recognize. And that oftentimes requires people getting out of their comfort zone, perhaps doing something that other people aren't doing, and making a new field for themselves, I think, is really, an important thing. And some of it at the educational level is designing your own education around what you want to do. And I think the days of just being like, I'm a biology major or a chemistry major, those days and those limitations aren't really applicable anymore. People need to sort of, like, think of what they have, a vision of what they may want, where they want to get to in their career, problems they want to solve, and just go ahead and start thinking about how to design their curriculum around it. One thing, and even on the job work experience, seeking out guidance or mentorship from someone who may be totally outside your field, and learning what they do, and start bringing elements of their knowledge into your own workspace. Incredibly important.
Excellent messages. Joel, while we have you, I know, in addition to the work you do in J&J, and as I mentioned, the involvement in Project DataSphere, you also are out there doing a lot of public-facing stuff on the conference circuit. I think you were at the Immuno-Oncology 360 recently. What else is hot for you in 2023? Any presentations, conferences coming up, places that we can see you, meet you possibly, and listen to the stuff you're up to? Anything else that I did not touch on, please take the floor.
Yeah. Well, conferences, we very much we work closely with our oncology colleagues, and so we have a big presence at conferences like ASCO, ACR, and so it's critical that some of the science that we bring forward, it's a multidisciplinary team with the drug development teams. Janssen always has a big presence at those conferences. This year will be no exception. I think. And over time, I think what you'll see this year and beyond is a much bigger element of data science as part of the Janssen presence at these disease-focused conferences. So those are the best places to catch me. And I'd say that we'll have a significant part of our data science organization there, really representing Janssen as part of the oncology drug development efforts.
Excellent, really excellent. Awesome journey you've been on, Joel. I'm just so impressed by, again, the way, as you mentioned, you're bringing all these tools together to do what you're doing in the development of these therapeutics, and of course the companion diagnostics, and really just changing the oncology landscape. So I'm rooting you on. Obviously I'll continue to follow everything that you're about there at the organization. For everybody that's going to be listening to this particular episode of our show across the various podcast networks or watching on our YouTube channel, again, you've been listening to Joel Greshock, Vice President, Data Sciences Oncology at Janssen R&D, doing really amazing things to discover novel therapeutic compounds and pharmacotherapeutic interventions for areas of unmet medical need in the oncology space. Joel, I want to thank you for taking the time out of your schedule to come talk to us and educate us for a little while. Obviously, thank you for everything you're doing for patients. And as we like to say on our show here, thanks for helping to create a better tomorrow for a lot of people out there via what you do. Really great story.
Thanks, Sarah. My pleasure to be here.